I invite Orit Peleg from the University of Colorado, Boulder, from the USA, three-dimensional tracking insights into firefly behavior and conservation. And just so the green is okay when it's yellow. Be careful, breaking this.
Okay. Okay, sounds good. Is this working? Is the microphone working? Oh, it's have to switch. Right. Okay, how about now? Sounds like it's okay. Cool. Hi, everybody. As was mentioned, my name is Orit. I'm a newish assistant professor from Colorado. And this is my first time at the Firefly International Conference. I'm really excited to be here. I already learned so much from all of the amazing expertise in the room, and thank you so much for the organizers for giving me the opportunity to tell you about some of the work that we do in my lab. So I want to tell you about tracking. I thought that since this is the first time that I'm meeting some of you, this is not working. Okay, oh, here it is. Okay. Just to tell you a little bit of background about the kind of work that we do in my lab and where I'm coming from. So I'm trained as a physicist, and I'm really interested in complex systems. As we just heard, the last talk was very much in that spirit. I work on several different biological systems, starting on a molecular scale, to swarms of insects. Those are usually honeybees, at least in the past. And we use a lot of mathematical modelings coupled with experimental studies, behavioral studies, and we sort of iterate between the models and experiments until their underlying principles converge. And so we're trying to understand animal behavior in this context of collective behavior, large, dense aggregations, and how individuals communicate with each other in this context. As I mentioned, been doing this for a while with dung beetles and honeybees. I'm happy to chat about that if anybody's interested. But you might be wondering, what am I doing here? I'm a physicist who studied different systems. And actually some of you asked me, so I thought I would just spend a minute and tell you where this whole thing started. So when I was a physics undergrad, long time ago, I went to a class of dynamical systems and we used this textbook. And I pretty much owe everything to that book and its author, Steven Strogatz. It was a very heavy mathematical, physical class. We learned about how electrons synchronize in atoms in order to achieve superconductivity. And Steven Strogatz, I'm sure many of you know him, is such a good science communicator. He gave us this relatable example that we can maybe more, relate to, and that was Firefly communication. And to this day, I remember, going to this class and thinking that the same models that explain how electrons synchronize can also explain how eventually fireflies synchronize. And that problem has stayed kind of dormant at the back of my head until I opened up my own lab, and then I thought it was a really good opportunity to start working on that problem. Okay, so we saw a lot of beautiful models actually in the previous talk, so I'm very happy. Gonzalo gave this talk before me, so I don't have to explain everything. I just want to, my talk is actually going to be more about data acquisition, but I want to motivate that by an aspect of some of the models that are usually being used for explaining firefly behavior. And as we saw in the previous talk, we're usually thinking about that process as a group of oscillators. The fireflies kind of blink on and off, and we can think about that as an oscillator. From a physics perspective, an oscillator is a really simple process that basically just has a phase, so that's the y-axis here. As a function of time, it's producing this repeatable periodic behavior. And in most of these models, especially the models that were talked about in the textbook that I mentioned before, the way to get two agents that are oscillators to interact is through this element over here. This is the coupling constant, and then basically it couples all the phases of all the interacting agents. This is a very high-level description of that. The coupling element K can be many, many different things. It can be something similar to what we saw before in the previous talk. It can be something like a constant. It can be a function of the distance between the individuals, and you can sort of think about this as fireflies that are very close to each other, are more coupled than fireflies that are maybe further away from each other. So it can be a function of the distance. It can be a more complicated function of the distance. And actually, there's a whole zoo of models, literally. And I'm not showing this to scare you. I'm just showing you so that you get an idea of how many models are there. This is just taken from a recent review about the Kuromoto model, which is these kind of models that I just mentioned. And of course, every time you tweak the interaction a little bit, it's going to result in a different collective behavior. So that's kind of where my interest arises. And while there were, of course, studies, as mentioned before by Moisef and some of the others, we were really interested in capturing the spatio-temporal interaction and not just the temporal pattern of the synchronization. So that's the gap that we sort of aim to try to fill in. And me and my lab members basically went straight to the source in the Smoky Mountains, where there's very large swarms of Photinus carolinus that are the synchronous fireflies. As we saw in the previous talk, they have this pattern where they flash a couple times and then they go quiet for a while and flash again. And these days with fairly simple cameras, some of the cameras that were mentioned actually yesterday in Radim's talk, we can capture those flashes. And while these movies are not as beautiful as the movies of Radim, we can still get pretty good contrast between the flashes and the background, which means that we can threshold them. So we can basically find pixels that are higher value, and then get where the flashes were. So this is really just one camera view. But I'll get to two cameras, but I just want to show you the kind of data we can get from this. So we can get very quantitative data about the y-axis is flipped, but it says the number of flashes. I don't know what happened. Converting to PowerPoint. That's my guess. Okay, so, but anyway, this is the number of flashes as a function of time at two different points in the night. So the top plot is when we have low density of flashes. You can see that, there isn't really an apparent pattern. And then as the number of fireflies and the number of flashes increases, we're starting to see the burst structure that is characteristic for that species. And the nice thing, since we're getting so much data over the course of the night, we can actually characterize the transition, the onset point of synchronization as a function of the number of fireflies that we're seeing in our field of view. Okay, and there's more things. I'm going to keep it more concise and just say that, as I mentioned before, we were really interested in the spatial aspect of that behavior. And to do that, we basically need to use stereo vision, two cameras, and that's very similar to how our own depth estimation works. We have two eyes, so we can estimate the depth. On the left side, that's the more traditional way to record things in stereo. So we have two external cameras looking at the swarm from the outside. You can find tons of software online to do the reconstruction for you if you wanted to use that method. We chose something slightly different. So we use the method here on the right side, which shows we use GoPro cameras that have 360 field of view. That's nice because we can really put them at the center of the swarm and sort of get things from the perspective of an individual firefly as if it was right there down in the swarm. So it did require some new math to do the spatial reconstruction and the depth estimation, but we did that. This is published, and eventually we can get things like this. So this is one of the first 3D reconstructions of the flashes that we did with our data. I also want to point out that this is, we published the software, so it's free to use. Anybody who wants to is welcome. I know that there's at least one person in the crowd, maybe two, that are thinking about it, so please talk to me if you're interested. And from the perspective, going back to the mathematical behavior, mathematical models of behavior, we can now reconstruct spatiotemporal correlation functions. So here is an example of such a function. So on the y-axis, we have the distance between flashes. On the x-axis, we have the time. And we can get this very precise measurement of how the distance between fireflies affect their synchronization. And we can potentially also plug that back into some of these models. Okay, just a couple of quick things, other quick things that we can do with this method. Here we are actually recording a larger area, so it's about 30 meters in depth. We were able to capture some of the wave propagation that were reported decades ago, but in a more quantitative way, where in this case, the onset of a burst started close to the camera, which is around here at 0, 0, and it was moving away from the camera, and we can capture that with our cameras as well. We can also track individual fireflies by basically connecting the dots, since we know where they are in space and time. And we are starting now to kind of dig deeper and see what individual fireflies do, or smaller groups of fireflies do, and how that might lead to the synchronization behavior that we saw earlier. I'll just point out one interesting thing that we're not entirely sure how to account to, but what we notice is that when we take individual fireflies away from the swarm, they don't behave as good oscillators. Their period is not very precise and it's very variable. So we are now testing new mathematical models to try to account for that emergent periodicity that they have as a group. So everything I showed you was with Photinus carolinus. I'll just very briefly say that we also worked with Photinus snulli, thanks to the gracious help of Joe Cicero. We also work with Photeris frontalis that are, they have a continuous flash pattern. I'm not sure if my movie will play. It didn't, sorry. But they have a continuous flash pattern. And we can see this really interesting combination of a phase of the swarm that is synchronous, that is accompanied by a totally non-synchronous phase. For those of you who are familiar with these processes, it's called chimera states sometimes. Okay, so all of this is seems to be helpful for understanding firefly behavior, but then mostly through conversations with Sarah Lewis, we thought that maybe some of these methods to record fireflies could be useful for firefly conservation. And the idea is that we have this sort of passive way to record firefly flashes. There are a couple of steps that I'll go through in the next couple of slides after recording what we think is about building a database, and then come up with a machine learning code to classify the firefly flashes based on the recorded patterns. So just one quick comment about the method. Some of you, I don't know if any of you are using stereo vision, but the most annoying thing about stereo vision is calibration, because you have to synchronize the cameras both in time and space. And we found a way to not need that in our method. And this is only because we have very good contrast between the flashes of the fireflies in the background. So we can do things like this, like plot the number of flashes we have from one camera view as a function of time versus the other camera view, and basically align them until they match. So we don't need to synchronize them in time. Same thing spatially. So basically, anybody who want to use this method need to go out to the field, put two cameras, press record, and that's it. Okay, and then we're working on creating this database where we have accounted for behavioral variability of fireflies. These are 3 species that we have. And eventually, what we're trying to do now is work with machine learning to classify the firefly species based on their flash pattern. We did this theoretical exercise with data that was published by Stanger Hall here in the audience. The output of these methods is basically this kind of matrix where on the y-axis you see the true species, on the x-axis is the predicted species. Whenever we see elements on the diagonal, that means that we characterize it correctly. It works very well with these theoretical sequences. It doesn't do a very good job with actual measured, recorded firefly patterns if they're trained on the theoretical patterns. So that's why we need more data. And that's where I want to give a really big shout out to some amazing scientists and firefly enthusiasts who are helping us. They're volunteering. Basically, we're sending them a kit with 2 GoPros and a hard drive. They go out to the field, they don't need to do any calibration, just press record. And then this is from last week, I think, from Richard Joyce from the Cersei Society, who recorded some fireflies for us. So if you have fireflies in your backyard or in your field site and you're willing to help us, please let me know. I'll be very grateful, and hopefully this kind of database can eventually be useful for firefly conservation efforts. Okay, and I think I'll just thank everybody in my lab who works on the Firefly project, collaborators, especially the ones from the National Park Service at Congaree and Smoky Mountains funding, and thank you all so much for listening. I'm happy to take questions.
Thank you, very nice talk.
Thank you.
You showed us that from an asynchronous mess, we went into a synchronized business. What about coming later? Is it possible to mark, for instance, females? See how this mess goes onto a female. Is a female in the middle of the swarm, in advantage, or on the sides, big, small?
So what I showed was mostly the males. We don't really have control since these are experiments that are done outside, but we're doing some experiments this year in the field with an artificial female LED, and we can see how the males are we can see that they're attracted to her, and this seems to be a good method to ask or to answer some of the questions that were asked in the previous talk about how the female affect the behavior of the males, what is individuality in a swarm that is totally synchronized, and these sort of things.
Okay, thank you.
All right.
I have no question.
Okay.
Thanks for your nice talk and for this new methodology. Actually, I have a proposal for maybe our Croatian people here, because in the Balkans we have a synchronous Luciola species, and I'm convinced that it is present in Croatia. So maybe if they would like to volunteer to have better data on this Luciola species. We worked actually with Andrew Mosev.
Oh, great!
Years ago on a poster where he analyzed some footage from Greece. It is a very nice methodology to analyze the flashing behavior of that particle. So the males fly and have this, when they are in close contact, they really synchronize quite.
Yes.
A bit like Photuris, help me.
Frontalis?
Frontalis, yeah.
Yes, exactly. Exactly. And I just want to point out that the method doesn't require synchronization of the males. It would also work with any species of fireflies,
sure.
But yeah, it would be great to have another...
I'm very sorry, we don't have flashing species in Belgium, so I cannot help you. We have more like blue ghost one, but maybe you can ask something.
We have them in southern France, and that's one of the problems we have, knowing where a mill is going.
Yeah.
Is a flash, is it that one?
So
Could this system help with it?
Absolutely, yeah.
Then we are
can
candidating.
track, yes, as long as they, within a burst. So I'm happy to talk more about this and see if it works out for your system.
And if the interflash pulse is very long?
Then we lose track of them, unfortunately. Yeah, so it's going to be depending on the density, the ratio of the density, how fast the fireflies are moving, and what is the break between flashes. And we can try to assess it. There's one more question at the back there.
Thank you. Thank you for the talk. Could you standardize for volume or area that's being sampled? Do you know what that is?
Yeah, so technically yes, because we have the exact space, the spot that we see each flash. So if you wanted to only consider a particular volume, we can easily just throw out anything outside that volume. I would also point out that depending on the camera, you might actually be restricted to a specific volume in any case. For example, with the GoPros, they capture the fireflies species we work with up to about 5 meters away from the camera, and after that, the light is a little bit too dim to be properly captured. But yeah, technically, we can standardize the volume. Okay, thank you all.